Então a gente vai agora escutar mais duas falas remotas. O Chris Dick, que vai trazer a fala sobre mais de 30 anos da parcela Lecythidaceae, um resumo da pesquisa recente, que o Beto já tinha citado. Então, por favor, Chris, é com você. Não estou te vendo não, mas eu espero te ver em breve. Opa!
Okay, so thank you, Beto, for introducing phylogeny, so I don't have to. I'm going to be talking about the diversification of Amazonian trees and especially some insights from the Lecythidaceae plot. So this talk has some general evolutionary themes. The main question that's interesting to me is about Amazon tree diversity is why there are so many species. And as Beto mentioned, there are over 1,300 tree species in the plots in Manaus, in the forest geoplot. And we actually have a forest geoplot in Michigan, and we have 45 species. So you can imagine how I would be interested in knowing why there are so many species in tropical forests, especially when you compare it to Michigan. So some thematic questions. What are the geographic origins of Amazon tree diversity? In particular, when we focus on particular clades of trees. What are the drivers of speciation of Amazon trees? And what evolutionary processes contribute to ecological success in Amazon trees? The group that I've been focusing on for about 10 years now, out of collaboration with Scott Morai, is Lecythidaceae. Lecythidaceae, the Brazil nut family. It's pantropical, but it has one major clade in the neotropics called the Lecythidoideae. And that's where most of the species are. And the center of species diversity is in the Guiana Shield region and in the Manaus region. The taxonomy is pretty well known for the family, mainly because of the efforts of Scott Morai and Gillian Prance. And there are about 10 genera of Lecythidaceae in the Amazon basin. The most important in terms of numbers of trees and biomass are Eschweileria, which currently has about 100 species, and Lecythis, which is about 30 species. These can be enormous sized trees and typically are important in terms of biomass, especially the Eschweileria and Lecythis. So here is a phylogeny of the Lecythidaceae, and what this shows is that the neotropical Lecythidaceae are sister to the African clades. We know more or less what the branching relationships are among the different genera, except when we get into this clade called the Bertholletia clade. And the Bertholletia clade contains Bertholletia, the Brazil nut, Lecythis, Corythophora, and Eschweileria. And here we really don't know what the relationships are. The whole family goes back in the neotropics about 46 to 56 million years ago. And this Bertholletia clade contains the most diverse genera. Okay. So I'm sure all of you are familiar with the Lecythidaceae. When I was a estagiario in the PDFF project back in the early 1990s, I was intrigued by the Lecythidaceae, and at that time, Scott Morai was out at kilometer 41, and along with interns, and I was able to see the full diversity of fruit types, and it was an incredible transformative experience for me. But more generally, the Lecythidaceae are known for horticulture. Here we have the cannonball tree. They're known for their timber, especially in the genus Cariniana. And then, of course, the Brazil nuts, Bertholletia excelsa. The Lecythidaceae have these very distinctive woody capsules. They have edible seeds, and they tend to be slow-growing trees of mature rainforest. They have at least one part of the family has these unique nautilus-shaped flowers that are specialized for pollination, typically by very large bees like euglossian bees or carpenter bees. And the seeds are consumed by rodents, bats, and as well as humans. Lecythidaceae has a large number of hyperdominant species that Beto mentioned, the hyperdominance. And so there are 16 species just within this Bertholletia clade that are considered hyperdominant. So these are among the 200 most abundant tree species out of at least 5,000 known species in the Amazon forests. And the one species, Eschweiler coriaceae, is estimated to have about 5 billion trees greater than 10 centimeters in dBH. So this contributes in an enormous way to the biomass of Amazon forests, to carbon cycling. However, this Eschweiler coriaceae may contain cryptic species, and this is something that we're studying. And Priscilla Souza, who's a PhD student at INPA, is studying this problem right now. So there are big identification challenges with Lecythidaceae, probably not as much as in some other groups like the Myrtaceae, but for many of these forest inventory plots, the identification is only at the level of genus. The reason being that oftentimes people only can identify the species based on flower morphology, and it's very difficult to get flowers. And in some cases, in the kilometer 41 plot, people have tracked individual trees for decades and still have not seen flowers from some of the unidentified species. Here are some recent discoveries. There are a lot of new species to be discovered and that are described every year. This it would seem like this would not go unnoticed. This is a tree with an enormous leaf in the genus Grius. It's known only from the cloud forest in Colombia, and the type specimen was collected just in 2009. So some more specific questions. We wanted to know what are the branching relationships, the evolutionary relationships among the major clades of the Lecythidaceae. Then we wanted to know about hybridization. So does hybridization play a role in generating genetic diversity in some of the hyperdominant species? And then how well does the taxonomy reflect evolutionary relationships? And this is a question is along the lines of also what Beto is interested in and discussed. So I began working on Lecythidaceae as part of the Dimensions of Biodiversity National Science Foundation project, and in collaboration with Scott Morai. And I was really excited to be able to return to the kilometer 41 plot, and I hadn't seen Scott there for 30 years. The kilometer 41 plot was established in the late 1980s by Scott and Peter Becker, who was an ecologist who worked a lot at the Forest Fragments Project early on. And so Scott considered himself to be a taxonomist and systematist, and he considered Peter Becker to be an ecologist. And what they wanted to do was combine and intensively study the ecology and taxonomy of a single family in the central Amazon basin to use it as a model system for both ecological and evolutionary studies. So at that time, they mapped all of the trees in 100 hectares in the kilometer 41 forest, everything above 10 centimeters. And so they ended up with about 7,800 Lecythidaceae trees. This resulted in about 40 species, including unidentified species. And all of these have excellent vouchers, most of them with flower and fruit specimens that are held in the New York Botanical Garden and the INPA herbaria. Since the late 1970s, over 13 new species of Lecythidaceae have been described, mostly from the kilometer 41 plot and elsewhere on the road between the Forest Fragments Project and Manaus. And there are at least 4 undescribed species of very large tree, and we have whole populations of them sitting out at kilometer 41 that Scott began to describe but refused to put a name on them until he could get a fruit or a flowering specimen. And people have periodically gone out and tried to collect over the past 30 years and have never returned with a fruit or a flower. So that's another issue with some of these taxa, is some of them are just not going to be producing flowers or fruits that you can use to identify them. I'll just mention also that there was a lot of additional data available for the plot, including soils data and phenology data, where people carefully mapped out the phenology of the species. And I could find none of that data. That data was apparently lost. So this is just another cautionary tale for how important it is to maintain data over time. The kilometer 41 plot is exceptional for studying evolutionary biology of trees, because, as I mentioned, all of the herbarium specimens that are, many of them digitized, and for these really important herbarium specimens where the species identifications or species descriptions have been made, they're associated with a tagged tree so that we can return to the same tree and continue to get data from that tree. So we can get transcriptomic data using RNA, or if it was collected in flower, we can go back and collect it in fruit. So that's really important. There's this really careful study, a monograph, mainly of the kilometer 41 trees, that was done by Scott and Nadja Lepkunya, which was very important for a newcomer to come in and understand the basic biology of these trees. And there's just an amazing international and local knowledge, unfortunately gone now, but to some degree, Scott having passed, but Paulo Apostolo was phenomenal in helping us in the field with a local knowledge and local detailed understanding of the trees. That was really important for us to get started. Let's see. So we decided to resurvey as part of our overall project. So, well, why not just recensus the entire plot? And we did some initial visits in 2017. And at first we couldn't find the plot. And Paulo said, I'm sorry, it must be just the tags must have fallen off. And then Paulo was finally able to find some trees with tags, and then we saw that everything was still intact. And so we began the process of recensus, and this was led by PhD student Tamara Milton. And part of her interest was in understanding the demography of the Lecythidaceae, and in particular, the flooding history and how some of the hyperdominants would respond to some of the disturbances in the plot. So, and what this shows are all the individual trees within the 100-hectare plot. And these are some of our key projects that we're working on. So PDBFF alumnus Bruno Garcia Luz accompanied me the first time to the plot along with Paulo, and that's when we first found these trees. We found an initial set and sort of established the old plot. We returned a year later and had additional help, and then this is when the recensus began in full scale. One thing that was really nice to work with Paulo and to learn from him about were all the distinctive field characters that we could use to identify species. So when flowers and fruits were not available, when the leaves were not distinctive enough or we couldn't reach them, there were characters like the slash, the inner bark, and these were consistently useful characters, and these would not have been available from your standard monograph. This was local knowledge for the most part. So we learned that the Lecythidaceae, I think we relearned or confirmed that Lecythidaceae grow very slowly. The average growth rate is about 1 millimeter per year, even for the canopy trees and the canopy emergence. This is averaged across trees. And I'm not a demographer, but this was surprising to me. And it made me think about sustainable logging that is based on a 30-year cutting cycle. Like, how useful can that be? And I realized that when you cut the forest, you potentially provide light and maybe the trees will grow faster. But this was really something to think about, was this slow growing forest. So a postdoc, Oscar Vargas, worked on developing genomic markers for the Lecythidaceae so that we could really work out at fine detail all of the phylogenetic relationships going between the genera and then within species. So he developed target sequence capture markers that had 324 loci. And this produced about 150,000 SNPs or polymorphic sites. And it ended up resolving just about everything with a few exceptions. And one thing we found, just looking at Eschweiler coriaceae, was that our various specimens of this species came out in different parts of the phylogeny. In one case, it was beside Eschweiler bracteosa, another case with Eschweiler wackenheimii, in another case, Eschweiler truncata. And this suggested to us that this hyperdominant species, ranked number 2 in terms of its overall abundance in the Amazon, was not what we would call a good species. And this suggested to us that there was a problem either with a misidentification or hybridization. And so to address this question, PhD student Drew Larson addressed the question about hybridization. And he went to the kilometer 41 plot and was able to go to these trees that had been collected previously. And even trees that we were able to identify in the field as having an intermediate morphology that could be hybrids. And he gathered enough specimens from enough individuals and used these target sequence capture markers and analyzed the gene pools using the program called Structure. And what he found was that most of the species in this Bertholletia clade, or the parvifolia clade within the Bertholletia clade, most species showed no evidence of admixture. So when you looked at what Scott Morai considered to be a good species, there was evidence from the genetics that they were good species. And what you see when there's something that has a single gene pool, you'll see just a single color. However, 3 species contained evidence of hybridization or admixture in both the phylogenetic and the structure analyses. And these are the 3 species. So you can see when there's been, looks like even a second generation hybrid, when for the single individual tree here on the bottom, single individual has a gene pool from 2 different species. And so that is very clear evidence that there has been some level of hybridization in these trees. So why is hybridization of these trees interesting? So what this does is this exposes the species to all kinds of gene combinations and adaptive variation through hybridization that can allow populations to acquire favorable alleles. And this may even explain why some of the hyperdominant species are ecological generalists. So for example, Eschweiler coriaceae can occur in flooded habitats, and it can occur in terra firme habitats. There may be other habitats throughout Amazonia where it occurs. Maybe it's able to jump onto these habitats through this process of acquiring parts of the genome of local species. Okay, the other thing that we wanted to do was look at the relationships of these genera, especially in this Bertholletia clade. And we actually confirmed what Scott Morai had suspected, which is what these 2 very important genera, Lecythidaceae and Eschweilera, are not monophyletic. So here we can see this very important genus Lecythidaceae. First of all, Bertholletia excelsa divides 2 major clades of Lecythidaceae. And then there's another group of Lecythidaceae that's very unrelated, and it has Eschweiler and Corythophora intervening. And Eschweilera also is not monophyletic. It occurs in different parts of the phylogeny. And so taxonomists really would like genera to reflect monophyletic groups, to represent clades and single lineages. So we're currently, we have a several-year project of redoing the taxonomy of the genus, of the family, and this is being led by Oscar Vargas. And so we're basically going to take these non-monophyletic groupings and then rename them. And so you can see what the new names are up there. So Lecythidaceae, this clade becomes Pachylecythis, this clade becomes Cretoma, this clade of just a single species in the kilometer 41 plot has a new genus name, Wymeria. These are all tentative names, so they could change. So what it means basically is that with these new changes, there will not be any more Lecythidaceae in the Forest Fragments Project plots. It's all going to have different names. And that could be frustrating for some people, but it will reflect the evolutionary history a little bit better. And it's really interesting that, there is this genus, what we're proposing to be a new genus, but a very isolated species, Eschweiler amazoniciforma, which is so distant from any other species that had probably diverged from them, at least in the early Miocene, like close to 20 million years ago. So it's a distinct evolutionary lineage, and it's only been collected in Manaus, so it's endemic to the region, and it's most common in the forest fragments reserves. And it just makes one wonder how many other really distinctive evolutionary lineages that are out there that are just waiting to be encountered. So just to summarize a little bit of the scientific findings. So the slow growth of Lecythidaceae is a real challenge for any kind of sustainable logging policy. Second, hybridization of hyperdominant Amazon trees may provide a link between adaptive evolution and demographic success. Lecythidaceae and Eschweilera are not monophyletic, and that's going to lead to major taxonomic revisions of Amazonian clades. So some other thoughts, and I'll end with this slide. The Lecythidaceae plot was really invaluable for the evolutionary studies that we did because of all of these years of it being studied intensively and the good documentation, at least of the taxonomy components, including the herbarium specimens and all of the tagged trees. And, that being said, valuable data from the plot has disappeared. And so the soils data was, used to be managed by Peter Becker, and phenology data that represents hundreds and hundreds of hours of work, and that's gone. And the local knowledge of the plot was critical for the success of this research. And I hope that there's opportunities to continue to develop that human resource aspect of the forest fragments plots. Also, it's possible to do a similar kind of study with the Sapitaceae plot. So there's a 50-hectare plot of Sapitaceae that's nested within the Lecythidaceae plot that has also been studied intensively, but not visited after many decades. Thank you. There are many people to thank, and sorry if I missed putting anybody's name up.
Já chamamos você de novo para a plenária de discussão e as perguntas. Muito obrigado.